Assalamualaikum and greetings to Madam Haniza, our lecturer. So for today, we are going to present our insect collection presentation for our entomology project. Before that, I would like to introduce my group members. So the first one is Aishah binti Mustapha Kamil, and then followed by me, Fathen binti Mohamad, and the last one is Hasveini, anak perempuan M.Kalai Chelvan. Moving on to the introduction.
So the introduction is that insects are collected from all habitats and for all purposes in order to provide information on their behavior and ecological requirements. There are two categories that can be applied to insect collection techniques. The first step in the collecting process is for a collector to aggressively seek for and gather the insects using nets, aspirators, fitting sheets, or any other tools that best matches their needs. The second step is the collector takes a passive role and lets traps do all the work. In this project, we mainly use the first method. This project focuses on to collecting arthropods. This is due to phylum Arthropoda being the largest phylum in the animal kingdom. Members of this phylum make up about 84% of all known animal species. Arthropods are prevalent in every ecosystem on Earth and exhibit a wide range of adaptations. Diverse species can be found living in terrestrial or aquatic environments, and some have even evolved the ability to fly. Now, moving on to objectives.
So the objective are to identify the collecting method that are appropriate for each species, to investigate the different type of arthropod species, to determine the proper pinning and marking technique for insect specimen, and to observe the main features of insect specimen. So moving on.
So the materials that we use is the fishing net, small plastic container, gloves, plastic forceps, pins, polystyrene board, plastic box container, naphthalene balls, and glue trap. The methods that we use to collect the insect is the sticky trap and the netting. The methods to kill the insect, we use the killing jar, where we use a makeshift small plastic box, and we put in the insect and we put in the naphthalene ball, slowly killing the insect with the naphthalene ball. And the second method is freezing. Now moving on to the results.
Okay, so as for the results, the first insects that we managed to caught for this one is the lesser banded hornet species. So based on the taxonomic classification, the lesser banded hornet species is the Vespa affinis. So the habitats is that it is mainly found on the tropical and subtropical regions of Asia. And then they nest above ground on a variety of supports such as trees, bushes, shrubs, and even buildings. And they are more commonly found close to the ground in grassy areas, forests, or wasteland. As for the life cycle, this Vespa affinis undergoes complete metamorphosis, which includes egg, larva, pupa, and adult stage. As for the medical importance, someone who might bite by the lesser banded hornet will cause severe anaphylactic shock, allergic reactions that caused by histamine, and then swelling and blisters, which can be treated by using corticosteroid and antihistamine medication. As for the collection method, the insect was caught by using a net and then was transferred into an enclosed container, and a naphthalene was added into the container. And then for the next insects is the black carpenter ant. For black carpenter ant, the taxonomic classification shows that the species is from Camponotus pennsylvanicus. So the habitat is that it lives in colonies with workers and a queen. Carpenter ants naturally nest in a deadlocks, hollow trees, and wood in forests, and then they chew a wood to build a nest cavities and tunnels, and this action helps in decomposition and breaking apart the decaying logs. As for the life cycle, they undergo complete metamorphosis. And then as for the medical importance, this black carpenter ant doesn't give any severe vector disease, just a normal disease such as swelling, itchiness, and minor irritations. As for the collection method, it is the same as what we use to caught the bees. And then next. For the American cockroach, the species came from Periplaneta americana. And then as for the habitats, the American cockroach are found in most shady areas such as hollow trees, wood piles, and marsh. They are occasionally found under roof shingles and in attics. And then these cockroaches dwell outside, but then it will wander indoors to search for food and water to avoid any extreme weather conditions. As for the life cycle, it's a bit different because this American cockroach undergo incomplete metamorphosis, which is hemi-metabolous life cycle, which includes only three stages, which is egg, nymph, and adult stage. As for the medical importance, it spread diseases through droppings, vomit, and mechanical transfer from their feet and other body parts. And of course, it transmits various diseases such as E. coli infections, cholera, fever, asthma, and food poisoning. As for the collection method, we use a sticky trap and then we transfer the cockroach into an enclosed container and naphthalene was added. And next, for the next is the Pennsylvania wood cockroach. So it doesn't make any difference with the American cockroach except for the species. This one is the Parcoblata pennsylvanica. Life cycle is the same as the American cockroach, but then for the habitats, wood cockroach was found in forest leaf litter, under logs and other damp outdoor areas with a lot of decaying organic materials. And then they prefer moist and damp natural habitats. But then for medical importance, this wood cockroach was not known the vectors of disease because they do not bite or sting human, but then their dropping, saliva, and shed skin can give allergies. As for the collection method, it's the same as the American cockroach. And then the next one is the Malay viscon, which is a butterfly. So as for the Malay viscon, the taxonomic classification, the species is Thanesia pelea. So they are mainly found in the nature reserve, but at times adults can be seen flying in other forested areas. And then both male and female have the habit of resting on perches with wings open and visiting the flowers or ripened fruits on the flowering or fruiting trees in their habitat. As for the life cycle, as we know, butterfly undergo a complete metamorphosis, which include egg, larva, pupa, and adult stage. And then for the medical importance, this species, they can't bite or harm humans because they do not have stingers, spines, fangs, or piercing mouth parts. So they were unable to inject any toxins into an enemy. As for the collection method, we use a net, we transfer into a container and naphthalene was added into the container. And then the next species.
So the next species is the giant brown mantis. So the medical importance of this giant brown mantis is where they can bite if disturbed and they can offer little harm to the people. So the collection method here is the netting and trapping. And then the habitat is where the giant brown mantis is found, mainly in southern and eastern Africa, where it lives in variety of environments, including forest, grassland, and much more. And then the giant brown mantis goes through an incomplete metamorphosis where it will pass through three separate phases such as egg, nymph, and adult. According to the taxonomy classification, the scientific name of this the giant brown mantis is where it is Archimantis stella. So those are the taxonomy classification. So next is Asian amberwing. So it's basically a type of dragonfly. So the scientific name of this Asian amberwing is where Brachythermis contaminata. So it's found in Asian tropical and subtropical climates, which its range including India, Thailand, Malaysia, and Indonesia. It lives in a variety of settings, including ponds, lakes, marsh, and slow-moving rivers. So this species is basically frequently seen around the vegetation-rich sources of water. So this species is undergo a complete metamorphosis, which is processing it through 4 stages, such as egg, nymph, and emergence, and adult. So this species is actually not harmful for the human, but it is actually a healthy insect that help in reducing of mosquito or other flying insects that may be of medical value as disease carrier. So this collection method is netting and trapping as previous one, and those are the taxonomic classification. And next, so then the next species is known as Chalky patcher, and the scientific name of this species is Diplocodes trivialis, and it found in a variety of wetland settings. They also live near rice field, waterways, and so on. This species prefers environment with aquatic plant and open area where it can rest and hunt. And the life cycle and the medical importance collection methods all are similar to the previous one, which is Asian amberwing. And those are the taxonomy of classification. Next is next is the or or sorry or Weber spider. So this the scientific name of this species is Avernus ventricosus. So it found in variety of environment across the world, including the forest, garden, even the urban area. They often form their web in areas where there are a lot of insect activity, like the near the plant or tree and outside lighting for the greatest prey capture. And it consists of variety stages such as egg, spiderling, juvenile, and adult. So these species have venom that may immobilize the victim, and they are usually thought to be safe for the human because the venom is mostly used to control the little insects. And the collection method here is web collection and pitfall trap. And those are the taxonomic classification of that species. So next is the spurtoriated grasshopper. So this is basically a grasshopper that the scientific name of this species is Melanopus mexicanus. So this grasshopper might be bite when touched or disturbed by the human, and they are not poison and do not transfer any illness to the people. But this grasshopper population can achieve greater quantities under certain conditions and force agriculture armed by consuming the crop and plants. So the collection method is net and pitfall traps. So it lives in variety of environment, including grass, land, agriculture field, and open place with dense vegetation. And the life cycle is where it goes through a life cycle consists of several stages, such as egg, nymph, and adult. So those are the taxonomic classification. And next.
So the next insect is the black-head antlion. The scientific name is Myamelon formicarius. So the medical importance for the black-head antlion is the antlion extract has the ability to both lower blood sugar levels and heal tissue damage brought on by hyperglycemia. A study found that antlion extract was efficient in decreasing blood glucose levels, and a different study shows that antlion larvae contain a substance that has glucosidase inhibitory action. So the collection method is the netting. The habitats of the antlions is that the antlions live in a range of usually dry habitats, including open woodland floors, scrub clan dunes, hedge spaces, river banks, road verges, under raised buildings, and in vacant lots. The life cycle for the blackhead antlion is that it goes through a life cycle consisting of several stages, which is the egg, larva, cocoon, and adult. So the last insect is the May beetle. The scientific name for the May beetle is Phylophaga species. So the medical importance for the of the May beetle is that the adults reportedly defoliate host plants, including pines, pecans, other hardwoods, and various wood crops. The immatures live in the soil and feed in the root zone, harming new tree plantings and row crops and pasture and turf grasses. So the collection method is also netting, and the habitat it lives in the soil and feed on plant roots, especially those of grasses and cereals. So the life cycle for the May beetle is that it goes through a life cycle consisting of several stages, which is egg, larva, pupa, and adult. And that's it for the insect. Moving on to discussion.
So for the discussion, the first objective has been achieved, where the study of collecting method of different species is essential for the entomologists to gather the specimen for scientific examination and analysis. So each species may require specific techniques and tools to capture them effectively while reducing harm to the specimens and their habitats. So this is where from this from this project, we get familiar with few techniques or collecting techniques such as pitfall traps or light traps. This is where in this pitfall trap is where you have to put the container on the ground to capture to collect the insect that crawl or walk. So this kind of collecting method that has been used in this project. And then the second objective is where also has been achieved, which is investigating different type of arthropod species is crucial for understanding the diversity, ecological growth, and adaptation within the arthropod, and phylum. So each group has unique characteristic behaviors and ecological interaction. So by investigating the different type of arthropod species, entomologists can gain insights into several topics such as the taxonomy classification and the behavior of the insect. So from that, the entomologists can able to identify the species. They can identify the each and every species, and they also know and differentiate accordingly. Moving on to the next.
Okay, as for the third discussion is associated with our third objective, which is we able to experience new knowledge of the correct pinning and mounting techniques for those insect specimens. So some aspects need to be considered when mounting, such as the type of needle, size of a needle, size of the insect, and the level of the insect's fragility to avoid any body parts of the insects from being broken. And then for the next one, we managed to learn ways to preserve and maintain the structure of insect specimen in order to avoid any parts of the insect's body parts from broken, as I mentioned just now. And then the fourth one is once the mounting process was done, our fourth objective was managed to be achieved, which is to observe the main features of the insect specimens. This is one of the crucial part of our project, since the morphology of each insect were able to be identified and differentiated with its relation on their habitats and life cycle. Okay, and then as for our conclusion, we managed to find the strengths and the limitations for our project, which is the strength of this project is that we're able to achieve all of the four objectives that we had stated for this insect collection project. And then we also managed to collect a total of 12 species of arthropods and able to identify every each of them based on what had been learned throughout the learning session in the class. And then as for the limitation, so the first one is inappropriate method of collection, because some of the insect that was collected, as an example, the Malay viscount got tear part of the wing during the process of catching by using a net. And then as for the improvisation, we would like to improve the quality of the materials, which is the materials that will be used to catch the insects, such as the net should be made of high quality material so that it will not disrupt any body parts of the insects. And then the second limitation is lack of preservation technique. Body part of insect were accidentally dislocated during the mounting and pinning process because they were too fragile and dry since they were not preserved properly. And then as for the improvisation, students should make further research on how to preserve the insects, because there is a proper way to preserve insects, which is to soak in a warm water to moisten them before the pinning process. So more reading should have been made before proceed with the mounting and pinning process. And then the next one is the appendices showing the insect collection box kit that we have submitted. And then references. Okay, and then that's the end of our presentation. Thank you.